Good morning, everyone. Good morning. Good to see everybody.
Good morning.
Today. Go ahead and type your name or your nickname into chat for me. Thank you. Very good. So this week in lab, we have two subjects we're going to be talking about. We're going to be talking about how we examine the growth requirements for certain bacteria in terms of their oxygen needs. And we're also going to look at how we can use ultraviolet light to control the growth of microbes. So those topics, both the growth and controlling growth, are on our schedule. Today, we're going to start into the topic of oxygen and growth. And that'll be what we're focused on today, although we won't be able to get all the way through the material today. Before I begin, does anybody have any questions about anything? Any questions about anything you've been seeing in lecture, or any questions about any of the quizzes you've taken, or lab questions? Anything at all? Remember, in terms of what's coming up for us on our schedule, let me pull my screen up for you. What you should see on your screen now is our week 6 module in Blackboard. This is our content folder for week 6. You can see there's a bunch of different things in it, including lecture quizzes for growth and controlling growth, which are our 2 lecture topics for the week. And then again, there are lab questions as usual that you'll be able to complete once we're finished with our lab this week on Friday. Looking ahead to next week, I'm just going to click on week 7. Next week is going to be an unusual week for us because you'll be having both a lecture exam on Wednesday next week, and you'll be taking a lab practical exam on Friday next week. So you'll want to make sure that both of those things are up on your calendar. That's coming up next week, and we'll talk more about that later on in the week. But today we're focused on our lab topic, which is oxygen and growth. So I'm going to go ahead and open up our slide set for the lab. You should see on your screen now a slide that says Lab Oxygen and Growth. And this slide talks about our objectives for the exercise today. So the first thing we're going to do is talk about the 5 different categories of relationship that different bacteria have with oxygen. We can put bacteria into one of these 5 categories based on whether or not they require oxygen, whether oxygen is toxic to them, or whether they're somewhere in between. Because there are some microbes that don't use oxygen as their final electron acceptor. Remember, we talked about that on Friday. There are some microbes that don't use oxygen, but oxygen is not toxic to them. They have no problem being around oxygen. And there are still other microbes that do require oxygen, but they don't require it in the full amount that they would be exposed to in air. So quick question for the chat. How much oxygen is in room air as a percentage? The air that you and I breathe, how much of that percentage-wise is oxygen gas? Does anybody know? What is air in terms of oxygen? So I've got Amelia saying 21%, Sean Posner saying 21%, Brooks saying 15 to 20. You're right on at about 21. It's a little over 21 actually, but so approximately 20%, 1/5 of air is made out of oxygen or contains oxygen. Do you know what the primary element in air is? What element makes up most of the air we breathe? You got it, Brooke. Amelia. Yep, it's nitrogen. It seems strange, doesn't it? We don't think of air as containing nitrogen, and we don't think of it as being mostly nitrogen, right? Most people think about air and they think about oxygen because it's the oxygen gas that we are so dependent upon, right? We have to have oxygen or we won't survive. So we would fall into the same category that those bacteria fall into that absolutely require it and that will die without it. Good. So the list of groups is right up here for you. Obligate aerobes, obligate anaerobes, aerotolerant anaerobes, micro aerophiles, and facultative anaerobes. So we'll define each one of these categories today. And the other thing we're going to do is talk about a special kind of growth medium, a kind of culture medium that we use specifically to test what category a microbe falls into. And again, we're talking primarily about bacteria. This type of medium is called thioglycolate medium. And this is an unusual medium. It's in a test tube, but it's slightly thickened. You know, you could call it a semi-solid medium. And when microbes grow in it, again, primarily bacteria, when they grow in it, they grow in a particular pattern depending on what their oxygen requirements are. So we'll talk about thioglycolate and how we use it. We're also going to talk about specific methods that we use to culture anaerobes. Now, an anaerobe is a microbe that is not using oxygen. Some of them find oxygen toxic, in fact. We're also going to talk about a test called the Catalase Test. And the Catalase Test is related to oxygen in that this is an enzyme that helps break down one of the byproducts of being exposed to oxygen. Oxygen is actually a molecule that you don't want to be exposed to in large amounts. You and I occasionally have to be put on pure oxygen gas when we are not well, when we're in the hospital or we're having some kind of health crisis. You know, you get a mask on your face and it's got pure oxygen gas for you to breathe. But we don't keep people on pure oxygen gas because oxygen has problems associated with it in our cells. We require it, but we also recognize that at large amounts, at large concentrations, certain byproducts can be produced. So we're going to talk about this enzyme catalase because it helps get rid of one of those byproducts of oxygen. We'll talk about what it breaks down. We'll talk about how we test for catalase. And we'll talk about how we can read the test, positive versus negative. And then finally, just like we did last week with special media, we'll perform an experiment together where we will use the thioglycolate medium to try to identify what oxygen category a couple of different microbes belong to. So I'm just going to review a couple of the things that I talked to you about on Friday regarding oxygen and why oxygen is required by some life forms on planet Earth. We know that all living cells, all of them, require a constant ongoing supply of this molecule called ATP. And remember, ATP stands for something. It's adenosine triphosphate. Cells use this molecule to temporarily store energy. So this molecule in a cell can be broken apart to release energy. We've talked about this. We've talked about how everywhere that you see two atoms bound together in a molecule, everywhere you see one of these straight, solid black lines, you know that two atoms have been covalently bound to each other. And we also know that all a covalent bond is a pair of electrons that are being shared by those two atoms. So for example, I can see a P right here, a phosphorus atom, and I can see an oxygen atom next door, and there's this straight black line between them. That tells me that this phosphorus and this oxygen are sharing a pair of electrons in their valence shells. Now, if I were to break that bond, I would release those electrons and along with them, their energy. And it's the energy that cells use to do work, to build things and break things apart every day. So ATP is the molecule that living cells use for this purpose. And every living cell has a need for it. Absolutely. There is no survival without ATP. One of the biggest problems, if you will, for cells is that they can't store it in large amounts. Most microbes, for example, might have a minute or two of ATP available to them. But if they can't continue to make it, they will quickly run out of that ATP and die. And human beings are the same way, as are all the other animals and all the other organisms that live on this planet. We have a few minutes worth at any one time, but once that ATP is gone because it's been used up, that organism either needs to make fresh ATP or they're going to die. So no surprise that we understand that there are different metabolic pathways available to cells to make ATP. We talked very briefly on Friday about how human beings and other animals use a pathway called cellular respiration to make their ATP. Now, there are actually two different types of cellular respiration. There's an aerobic form. And anytime you see that word aerobic, think oxygen. And there's also an anaerobic form of cellular respiration. They are both cellular respiration pathways, but one of them requires oxygen and the other pathway doesn't. It requires a different molecule to be the final electron acceptor. Now, there is a third metabolic pathway that some organisms use to make ATP, and that's fermentation. Microbes are a lot more diverse than human beings are in terms of their metabolic abilities. Humans use aerobic cellular respiration to make their ATP. That is their primary metabolic pathway for making ATP. Some microbes also use aerobic cellular respiration, but other microbes use anaerobic, and some microbes only use fermentation. There are still other microbes that use a mix of these. In other words, they can do different pathways under different circumstances. So they're a lot more metabolically diverse than you and I are. So one of the ways that we identify microbes in the microbiology lab is by testing what kind of metabolic pathways they use. And one of the things we can test is for their oxygen requirements. Once we know that we're dealing with an organism that falls into one of those 5 oxygen requirement categories, once we know what it prefers, the next time we're trying to grow that microbe in the lab, we can adjust our culture conditions. In other words, there are ways that we can remove oxygen from a chamber, and that will allow us to grow any microbe that finds oxygen toxic. We can reduce the amount of oxygen in a particular culture environment for organisms that prefer a lower percentage than what's in the air. So we can make all kinds of adjustments once we know what a particular bacterium likes. So let's talk through these 5 categories of oxygen requirements. First on the list is the obligate aerobe. So remember, obligate meaning absolutely necessary. And when you see the word aerobe, I want you to think oxygen. So an obligate aerobe has an absolute requirement for oxygen. And that's because they use oxygen. They require oxygen to make their ATP. Remember, cellular respiration is a long, long series of chemical reactions. And just before the ATP is made, we're at the end of this structure called the electron transport chain. Oxygen sits at the bottom of that chain, and oxygen functions to accept electrons that have been removed from glucose. So the oxygen binds with, reacts with the electrons, as well as some hydrogen, and creates water. If there's no oxygen around, that whole system shuts down and the organism dies. Obligate aerobes use aerobic cellular respiration. Some of them can also use fermentation. That varies by species. And the catalase enzyme, is usually present in the cells of an obligate aerobe because they're using oxygen. There are also oxygen byproducts found in the cell, and they can be toxic. So catalase is the enzyme that helps break those byproducts down. Now, on sort of the opposite end of the spectrum, there's the obligate anaerobe. This is an organism that requires no oxygen, anaerobe. So not only can they not use oxygen to make ATP, but they find oxygen toxic. They're going to use cellular respiration, but they're going to use anaerobic cellular respiration. They're going to use a molecule other than oxygen at the end of the electron transport chain. Some of these can also use fermentation. Again, that varies by species. These organisms typically don't have catalase enzyme in their cells because they really don't need it. Remember, catalase is there to break down one of the toxic byproducts of oxygen exposure. So if oxygen is toxic to you and you don't need it and you don't tend to have it in your cells, you don't need this enzyme around. Next on the list is a group called Tolerant Anaerobes. So again, the anaerobe tells you no oxygen, but they're also aerotolerant. So these are organisms that don't use oxygen to make ATP, but to them, oxygen is not toxic. They can tolerate oxygen. They're not going to use it, but it's not going to kill them. These organisms typically lack catalase, but they also don't tend to have the problem of generating toxic byproducts from oxygen. They just don't tend to make toxic byproducts from oxygen. They're not using oxygen, and they just don't have a need for the enzyme systems to break down toxic byproducts. I have one of those byproducts listed here, H2O2, which is hydrogen peroxide. Aerotolerant anaerobes also have the ability sometimes to use fermentation. Aerotolerants, in fact, are more likely to also use fermentation. Remember, so far we've said any of these groups. Some species might also be able to ferment. The Aero tolerants, they can usually ferment as well. Now, here's an important point to know about Aero tolerant anaerobes. They're going to grow best in low concentrations of oxygen. Now, that might seem very strange because they're not using the oxygen to make ATP. So why in the world do they grow best in low oxygen concentrations? That's not something I can explain chemically to you. They will grow in a condition where there's no oxygen. They will grow when they're in room air. But they grow best when there's a low level of oxygen present. They're not using it to make ATP, but for some reason, metabolically, they prefer to have a little oxygen around. So those are the first three categories. Let's take a look at what that looks like in a thioglycolate tube. And that's what you're looking at on this slide. You're looking at 5 test tubes that are filled with this sort of beige colored liquid. This is thioglycolate. And remember, I told you a minute ago that thioglycolate is a medium that we use specifically to determine what kind of oxygen requirements a microbe has. They're going to grow in this tube in different patterns depending on their oxygen requirements. This medium is not solid like agar, but it's also not liquid like broth. It's halfway between the two. It's got a little bit of agar in it, so it's sort of semi-solid. Now, what you're looking at in this first tube labeled A, you can see the thioglycolate medium, the beige colored material, and then you can see these red dots at the top. The red dots represent the bacteria growing in the tube. So as you can see, it's growing only in this upper portion of the tube, maybe about the upper 1/5 of the tube, the upper 20% of the tube. These are obligate aerobes, the organisms that grow in this pattern, way up at the top like this. Those would be obligate aerobes. This top 20% of the tube has oxygen in it. And the reason it has oxygen in it is because this part of the tube is closest to the air. So the air can interact with the glycolate medium right here, and some of the oxygen will dissolve into the medium. Now, it can't get down very deep, but it can go down to this level. So we give this area of the tube a name. The top 1/5 of the tube, the top approximately 20% of the tube, is what we call the Oxic Zone. And it's got oxygen in it. So obligate aerobes have an absolute requirement for oxygen. No surprise, they're going to be limited to growing in that oxic zone. As you go down into the deeper parts of the tube, there's less and less and less oxygen down here. And when you get down to the lowest 20%, there's no oxygen. So we can refer to that as the anoxic zone, no oxygen. Now look at how an obligate anaerobe grows in a thioglycolate tube. They're going to grow down in the lowest part of the tube because there's no oxygen down here. Remember, obligate anaerobes find oxygen toxic, so they have to stay away from it or they won't survive. So they go way down to the very bottom part of the tube where there's no oxygen, and they can grow right there. Now you have to jump over this tube for a minute and look at this one. This is the Aero tolerant anaerobes. Now there's a little green box right here. I'll explain that in just a minute. What I want you to notice is that there are red circles up and down this tube through most of it. And there's no one place that they tend to be growing. Yeah. In other words, they're dispersed throughout the tube. Remember, we said aero-tolerant anaerobes don't require oxygen, but they also don't mind it. It's not toxic to them. So they're able to grow in the presence of oxygen, and they're able to grow without oxygen. But remember, they grow best in low concentrations of oxygen. So I put this square here because in this source material, I'll move the square for a minute. In the place where I got this image, and I do like this image, the artist drew it as though there was no one place in the tube where they grew best. And that's just not what we see in real life. In real life, we will see growth throughout most of the tube, but most of the growth will be in the lower half of the oxic zone. So remember, the oxic zone is the top 20%. And these organisms like to grow in the lower half of the oxic zone. That's where they grow best. And they'll also grow in other parts of the tube. All right, let's look at these other two categories now. The fourth category is the micro aerophile. Now, if you translate this word, remember micro means mini or small or, microscopic for us. Aero refers to air, and phile or philic refers to love. So a micro aerophile likes a small amount of air. They love phile, a micro amount of air. And here we can read air as oxygen. These organisms do require oxygen to make ATP. So just like the obligate anaerobes, they have to have oxygen or they won't survive. But here's the important distinction. They require oxygen at a reduced concentration. Now, these organisms tend to grow best in low oxygen concentration and high CO2. So you can see I've written 5 to 10% CO2. That's more carbon dioxide than there is normally in air. We can set up one of those sort of specialized environments if we know we're trying to grow a micro aerophile. It's actually very low tech. You can take a jar with a tight fitting lid, and you can put your plate or your broth tube with the micro aerophile in it in the bottom of the jar, along with a candle. You light the candle, and then you close the jar. You put a lid on it and seal it up. What's going to happen is that candle is going to burn all of the oxygen. And then the candle's going to go out, right? Because there won't be any oxygen left. Now, this is not a method that will remove all of the oxygen, but most of it will be removed. This is not high tech enough to remove all of the oxygen, but it will remove most of the oxygen from that jar. So only a little bit will be left behind. And that's exactly what these organisms like. Now, there is a modern version of this CO2 jar, which actually generates a little bit of carbon dioxide as well as removing the oxygen with the candle. So there are different versions of this jar available, but what this jar is doing is creating a low oxygen, high CO2 environment for these organisms to grow in. Last on the list is a group called the Facultative Anaerobes. This word facultative tells us that these are organisms that can switch. They can switch very readily between metabolic pathways. They are anaerobes. Remember, that means they grow without oxygen. They can grow without oxygen. They don't require it, but they have the most diverse abilities of all of the groups we've talked about so far. A facultative anaerobe actually grows best with oxygen, and it's the full amount of oxygen that's in the air. So they grow best with oxygen, but they don't require it to make their ATP. They can also use other pathways to make their ATP. So they're capable of doing aerobic cellular respiration. They're also capable of using anaerobic cellular respiration and fermentation. All three pathways are available to this group. Here's an important thing to jot down in your notes and put a star next to. Most bacterial species are in this category. Most bacteria are facultative anaerobes. Not only can they use oxygen, but they can survive without it, and they can ferment. They have multiple pathways available to them in order to make their ATP. So when they find themselves in different conditions in the environment, they're able to switch. Very, very adaptive to be a facultative anaerobe. Now, if we come back to our diagrams here, this tube over here labeled E represents the micro aerophiles. Remember, these are organisms that require oxygen to make ATP, but they don't like the amount that's in room air. They prefer low concentrations, and they like a higher amount of CO2 than normal. So if you compare this tube to this tube marked A, the obligate aerobes can grow all the way through this oxic zone. But the micro aerophiles, they don't grow up near the surface of the medium. They grow in the lower half of the oxic zone. Now, if we look over at the facultative anaerobes, they can grow all the way through the oxic zone. In fact, they prefer the oxic zone. That's why you see most of the red dots up here. They can also grow without oxygen, though. And that's why you see some dots down through the rest of the tube as well. Now, if I were to fix the drawing of the Aero tolerance, if I could fix it, what I would do is I would take the micro aerophile and I would move it over here, because Aero tolerance prefer to grow right here in the lower half of the oxic zone, just like the micro aerophiles do. But Aero tolerance can also grow through the rest of the tube, just like the facultatives can. Now, one other thing to note about bacteria growing in thioglycolate, I wish that the actual tubes in real life, I wish that they grew exactly like those diagrams, but they don't. And part of the reason they don't look exactly like those diagrams is because bacterial cells tend to clump together when they're growing. So we will see something that looks similar to those patterns that I showed you. But instead of seeing cells nicely dispersed all the way through the tube, you're often going to see them growing as strings of cells. And that's because they're all clumping together in strings. And you'll see that when I show you the actual experimental tubes. All right, we're going to switch gears here for a minute and talk about this Catalase test. Remember, we said depending on which group a bacterium falls into, which oxygen requirement group it falls into, it may or may not have catalase available. We expect to see the catalase enzyme inside the cells of any organism that requires oxygen to make ATP. Any organism that requires oxygen is always going to have oxygen in their cells. And while that oxygen is helping make ATP, it's also being converted into these toxic byproducts. One of the most toxic is hydrogen peroxide. And hydrogen peroxide is toxic to living cells. Now, if you're an organism that doesn't use ATP, and let's say if you're an obligate anaerobe and you live in environments where there isn't any oxygen, you don't need catalase in your cells because you just don't get exposed to oxygen. Oxygen is just not in you because there's no need for it to be inside of you. So they don't have a need to break down the byproducts of oxygen. Now, what catalase does very specifically is it takes the hydrogen peroxide molecule, that's H2O2, and it breaks it into water and oxygen gas. So it's detoxifying hydrogen peroxide. This is one of the easiest tests to perform in the microbiology lab. Very, very quick, very easy, very inexpensive. What you do is you take a glass slide and you place a little bit of culture material in the center of the slide with your inoculating loop. You can take some cells out of a broth tube, or you can pick up some colonies off of an agar plate. Either way, you just want to spread the cells in the middle of your slide. Then you're just going to put a drop of hydrogen peroxide on top of those cells. So again, very easy to do this. What will happen if that cell has any catalase in it, I should say if those cells have any catalase enzyme in them, it's going to immediately start breaking down the hydrogen peroxide, and you're going to get bubbles forming. And these bubbles are this oxygen gas being let off as the catalase enzyme breaks down the hydrogen peroxide. So on this slide right here, you're looking at a catalase test, a positive versus a negative. This is a positive result for catalase. Obviously, this isn't a video, so you can't see this happening in real time, but it will literally just start to bubble up like this. I took some cells and I put them in the center of the slide. I just made a smear, and then I put a big drop, a big amount of hydrogen peroxide on top of those cells. And within seconds, you will see this bubbling. It starts to foam on the slide. Very, very easy to see it. Very easy to read this test. This is a positive result for catalase enzyme. Now, this is a negative result. In other words, if I put the hydrogen peroxide down and nothing happens, that's how I know that these cells don't have catalase enzyme. The hydrogen peroxide is just sitting there. So a very easy test to read, very fast. It happens immediately. You don't have to wait. You just take some cells, you make a smear, you put the hydrogen peroxide on top, and you watch for the bubbles. Now, here's a question for you. Human beings are obligate aerobes. We require oxygen. So no surprise, we also have catalase enzyme. So our cells can break down hydrogen peroxide. What is the organelle in your cells that stores catalase? What organelle do humans have in their cells that breaks down hydrogen peroxide? What's the name of that organelle? Go ahead and type it in the chat if you know. Bacteria don't have organelles, so their catalase is just sort of floating around in their cytoplasm. But you and I have organelles. And there's one organelle whose one of their main jobs is to break down hydrogen peroxide. That's the question, Brooke. Is it the lysosome or is it the peroxisome? The name kind of gives it away, right? It's the peroxisome. Peroxisomes break down hydrogen peroxide. They are where the catalase enzyme is stored in human cells and other animal cells. So one of the main jobs of a peroxisome is to break apart or detoxify hydrogen peroxide. And just like in the bacteria, it's broken into water and oxygen. Good job. All right, I want to take a few minutes and talk about some of these specialized environments that we can create in the laboratory if we're trying to grow an anaerobe, an organism that doesn't need oxygen and has other requirements. One of the most common containers that we use is called a Brewers jar. Here's the word right here. It's also known as a Brewer's Anaerobic jar. Again, it's just a big jar. It's a big plastic container with a tight fitting lid. Now, unlike the CO2 jar, the Brewer's jar, we have to remove every trace of oxygen. Remember, when we're growing an obligate anaerobe, the oxygen is actually toxic. So we got to get all of it out of this jar so that these cells can grow. And this is how that works. Inside the jar, when you buy one of these jars commercially, you get what's called a hydrogen gas packet. And these packets, they look like juice packets, you know, those juice containers that little kids use that have the aluminum foil kind of pouches. This comes with the kit, this hydrogen gas pack. What you do is you poke it, you open it, you put it in the jar, you put your plate in the jar that have cells on it or your broth tube, and then you put the lid on and close it up tight. That hydrogen that is being released from the gas pack is going to combine with oxygen in the jar and it's going to make water. And while this happens, you can watch the water will condense on the sides of the jar. These jars are transparent. You can see through them. So you know that your gas pack is working when you see this condensation, this water showing up on the sides of the jar. That's going to take all of the oxygen out of the brewer's jar, and any anaerobic, obligate anaerobe will be happy to grow in there for you. You take the whole jar and you put it in the incubator. You've created a little micro environment for those cells that has no oxygen in it. Now, the other way you can grow cells is with that thioglycolate broth, right? That thioglycolate medium. Remember, we said way down at the bottom of the tube, there's no oxygen. So it is possible to inoculate a thioglycolate tube, not so much because we want to see what category a microbe falls into, but we want to try to grow an anaerobic microbe. You can use these thioglycolate tubes for that as well. Let's talk just a little bit more about what's happening in that tube. I said earlier that we put a small amount of agar into the thioglycolate. And what the agar does is it makes the broth a little thicker than it normally would be. And that's going to reduce the ability of oxygen gas from the air to penetrate down into the broth. You're still going to get oxygen penetration up in the upper 20%. That's the Oxic Zone. But the further down the tube you go, you'll get less and less and less and less oxygen till there's no oxygen down at the bottom. Now, the thioglycolate, that's actually a molecule called sodium thioglycolate. And what this stuff does is combine with oxygen and help remove it, again, from the deepest areas of the tube. it is possible, I'm just going to mention this, it is possible to buy thioglycolate tubes that have already been made for you, so you don't have to make them yourself in the lab. And sometimes when we buy them, they have one of these indicator dyes added. Remember, indicator dyes are going to change color under different conditions. And this particular dye turns pink in the presence of oxygen. So you can buy thioglycolate tubes that will show you exactly where the oxic zone is in the tube, because the media will turn pink in that area. Now, a lot of people love that tube, like they love that version of the tube. It just makes it easier to read the tube. But you can also buy thioglycolate that doesn't have any indicator dye in it. And you just have to know that if anything grows up in the top 20% of the tube, that means that that's where the oxygen is. The third way that we can grow anaerobes, we have the Brewers Jar, we have thioglycolate, and we also have special anaerobic incubators on the market, as well as glove boxes. Now, anaerobic incubators are very expensive. If you think about it, this is an incubator that has to be able to keep oxygen out. So super expensive, tens of thousands of dollars for these. You won't find anaerobic incubators in most microbiology labs. It's just not worth the money. But if you were in a lab that grew anaerobes like as their primary work, maybe a research lab, they might have one of these incubators. It just depends on the work they're doing. The other thing is this device right here. This is one of the glove boxes. As you can see, this has holes in the side of it, this container, with gloves attached. And there's also this metal duct right here that you can attach tubing to. This is just, it's just a box. It's a plexiglass box with very tight fitting openings. So basically what you do is you attach tubing right here to the metal piece, and you can pump oxygen out of this environment. And the glove areas, these would allow you to reach into the box without opening the box. You stick your hands into these gloves and you can work in here. Now, I've been in labs that use glove boxes. I have worked in a micro lab that use glove boxes, but again, they're not very common. It's much more common to just use a brewer's jar when you need it. Brewer's jars are much less expensive. So they're much more common in the lab. They're very easy to use. They're very inexpensive, and they meet your needs most of the time. All right. So this is what a brewer's jar looks like up at the top here. You can see it's just it's a transparent jar. It happens to be made out of plexiglass kind of material. It has this lid with this thumb screw on top, and that allows you to put the lid on and really screw it down tight and make a very tight seal. Remember, we're trying to create an environment here that has absolutely no oxygen in it. Now you'll notice it's cylinder shaped, and that's because most of the time what we're going to be putting in here are agar plates. We're going to try to grow these anaerobic organisms on agar plates, and you just stack the plates up in here. When you buy your brewer's jar, it looks like this. It comes with these packets in it. The silver packet is the hydrogen gas packet. So I would put my agar plates in here first, and then I would take this silver packet, I would cut it open, place it in the jar, and then tightly screw down the lid. Remember, the hydrogen gas packet is going to remove the oxygen and convert it into water. You can also buy jars that have these white packets. Not all of them have these, but this is, again, an indicator dye. that will change color if any oxygen gets in here during the incubation period. So again, you put the silver packet in, you open it up, put the silver packet in, that's your hydrogen gas. You can also put one of these white packets in here. And if during the incubation process, any oxygen gets into this jar, maybe the seal isn't tight enough, or maybe it's broken somewhere, this packet will turn color. It's usually pink. So when I come back the next morning, if I see a pink color change here, I know, uh-oh, oxygen got in here. I probably was not successful growing my cells. Over here on the other side of the slide, this is a thioglycolate tube. Again, this is not a solid, but it's also not a pure liquid. It's somewhere in between. It's a semi-solid medium. This one doesn't have the indicator dye in it. This one does. This is just another tube, and you can see it's pink at the top. That's because there's a dye in here that will turn pink if it gets exposed to oxygen. So with the pink color change, I know exactly the border of the Oxic Zone. So when I put cells in here and I watch them grow, I can figure out, are they growing in the Oxic Zone? Are they growing outside of the Oxic Zone? The tube helps me determine that more easily. Okay, does anybody have any questions? Any questions about those 5 categories, first of all? The 5 oxygen requirement kind of categories: obligate aerobe, obligate anaerobe, aerotolerant, micro aerophile, and finally the facultative anaerobe. Remember, most bacteria fall in that last category. We will see species that fall into all categories, but most fall into the category of facultative anaerobe. All right, no questions. Very good. I'm going to go ahead now and talk about what our experiment is going to be for oxygen and growth. So for today's exercise, or actually we'll be talking about this more on Wednesday, the bacteria, or the bugs we're going to be using, as I like to call them, there are 4. The first one is called Clostridium Sporogenes. That's how you say that terrible looking word. Clostridium Sporogenes is a soil bacterium. It likes to live out in the environment. You can find it in regular soil, like garden soil, or you can also find it in the sediment in the ocean, so marine sort of sand and sediment. And it also grows inside the intestine, specifically in humans and other mammals. So this is an organism that you can find growing inside of animal, specifically mammalian intestines. Clostridium sporogenes is a gram-positive rod. This is what it happens to look like under the microscope. So gram-positive because it's purple, right? And if you look around for individual cells, you can see that they're rod-shaped. Here's a rod right here. Here's another one right here. These are rod-shaped cells. So Clostridium sporogenes. Remember too, Clostridium is one of the 2 genuses of bacteria, or genera of bacteria, as we say, that can sporulate. We said Bacillus and Clostridium are the 2 spore formers, just as an aside there. Second on the list is Enterococcus Fecalis. Now, we talked about Enterococcus in the special media lab. Remember, this is a gram-positive coccus, And it's also an intestinal organism or a fecal organism. You can find it in, again, the mammalian GI tract. You can also find Enterococcus in other parts of the body. You can find it in the female reproductive tract, at least in the lower parts. And you can even find it up in the higher parts of the digestive tract. You can find it in the mouth sometimes. This is Enterococcus. Take a look at these cells. If you look right here, for example, you can see two circles side by side, right? If I look over here, I also see two circular cells side by side. But then if you look in other places, like look right here, they almost look like rods, don't they? Enterococcus is one of the bacteria that have that unusual shape that we call a coccobacillus. So it's halfway between a coccus, a sphere, and a rod. If you remember from lecture, coccobacillus is not a shape we see very often, but Enterococcus happens to be one of those. Now, sometimes when you look up that organism in textbooks, it will be recorded as a gram-positive coccus, but other times you will see it listed as a coccobacillus, because it really does have that sort of in-between shape. So another gram-positive organism. Now, third on the list is our old friend E. coli. Remember, E. coli is a gram-negative rod. And we also find E. coli quite normally in the gastrointestinal tract. Very, very normal to find it there. Some strains of E. coli are pathogenic, but other strains are not. The last organism that we'll be using for this lab is called Alkalogenesis Facalis. That's how you pronounce that terrible looking word. Alkalogenesis Facalis. This one is also gram-negative and rod-shaped like E. coli is. And like the Clostridium, it can live out in the environment, in soils, in sediments, and it can also be found in the gastrointestinal tract. So we have two gram-positive organisms and we have two gram-negative organisms that we're working with today. And again, on Wednesday. Two of them only live in the intestine, or at least in the body. Two of them can live in the intestine. They can be found in the intestine, but they can also be found out in the world, out in certain environments. Now, before we started the experiment, I'll let you know that I confirmed that all of the cultures were alive and able to grow. So just like we saw last week, what I did was I grew these organisms in lab to make sure that they were alive and that they were able to grow. So we knew right at the beginning, at least, that we had healthy growing cells to work with. Now, here's the media we're using. We're going to use the Thioglycolate Broth because we're going to be interested in determining what oxygen category these four organisms belong to. We're also going to use nutrient agar plates. We're going to use a basal media. And the reason we're going to use those plates is so we can test to see if they can grow in a brewer's jar and if they can grow in room air. In other words, can they grow on nutrient agar exposed to oxygen, that 21% oxygen level? And can they grow in a brewer's jar with no oxygen? And the last thing we'll do is we'll perform Catalase testing. Now, I chose nutrient agar. Remember, this is a basal media, and that tells us something. That tells us we know all of the ingredients that are in it. and it will grow almost anything as long as it's not fastidious, as long as it doesn't have very particular nutritional requirements. And none of our organisms fall into the fastidious group. So this will be our experimental setup for Wednesday. We're going to take thioglycolate broth and we're going to put cells in. And then we're going to take those tubes, those 4 tubes, one for each of the organisms. We're going to put them in the incubator. I'm also going to spread each one of those organisms on its own nutrient agar plates, 2 plates per organism. One of the plates will just be left in the incubator. And one of the plates will be put into a brewer's jar and then put in the incubator. So we should have lots of different interesting looking results on Wednesday. And the last thing we'll do is we will take some of those cultured cells, put them on a glass slide, and put some hydrogen peroxide on top to see what we get, to see what their Catalase Test results look like. And once we have all of that on Wednesday, we'll be able to put each one of those organisms into one of the oxygen categories. So that'll be on Wednesday when we get together. Anybody have any questions? All right, very good. All right, I'm going to let you go. Enjoy the rest of the afternoon. It's a lovely day today. Enjoy the rest of the afternoon.
Thank you.
I'll see you on Wednesday. You're welcome, everybody.
Thank you.
Thank you.
You're welcome.